In this section, we are going to start focus on the interaction between multiple genes, and I will ask you a few questions about it. Okay, this is the fifth section. Here we're going to extend Mendelian genetics for 2 or more genes, and these are the questions 14, 15, and 16. So here will be the answers, and in the case, if you remember, there are qualitative characters and they can also be quantitative characters. So to say that something is tall and short, it will be qualitative, yeah. And to have a quantitative characters, we have to hide, for example, the height, and we will measure how tall and how short it is, and in that way we can define a quantitative in over a continuum. So here it will be the height in centimeters, meters, inches in any scale. So polygenic traits are usually measured in quantitative traits, in quantitative variation, and they seem to have an additive effect by more than one gene in one phenotype. And one example is the skin color. That is an example of a polygenic trait. In this section, we are just not going to talk more about polygenic traits or pleiotropy. We are going to start moving into epistasis. Remember the variation from the 9:3:3:1 ratio. So there are a few questions. Hopefully, if you have any questions, just let me know. Now we're going to talk about the epistasis. And epistasis is, here is the answer, and is when a gene at a locus alters the phenotype expression of a second locus. So in other words, that is an interaction between gene A and gene B for the production of a phenotype. And this is the thing that we talk in the classroom about the gene interaction. And in some cases, one gene can mask the action of the other one. So this is why we can get this 9:7 ratio instead of 9:3:1. Finally, in this last section, I will talk about X-linked traits, and I will give you a few more examples that we have a talk in the video lecture, and also I will add some examples of penetrance, some question about penetrance and multiple alleles that they can be in the test. If you have any questions, just let me know. So this is the seventh part of the assignment, and here we're talking about linked traits, and I'm asking you about this cross between a mother whose father was colorblind. So this is why you can see this allele, that is the allele for color blindness. And here you have that in the cross, you have to cross that woman that is heterozygous with this man that has a normal vision. So here, let me give you here you can find the male with a normal vision. Here you find the female with the colorblind allele. And when I fill the table, the Punnett square, you can see that you can find this case is a male that has this colorblind allele, while the other male has a normal vision. And in the female, you have females that are normal and another one that is a carrier. So you will find that they will 50% of the males will have a normal vision, 50% will be color blinded, and the females 50% will have a normal vision and 50% will be a carrier. When you have a red-green colorblind, this is an example of what? X-linked trait, Y trait, both X and Y, or none of these options. So definitely is not both and not none of these options. So it has to be one of these ones. So as we watched in the previous exercise, this is an example of a X-linked trait. And you will watch for the following lecture how Morgan found these linked traits. And we will watch a video about color blindness that describe other type of linked traits that are related to the X chromosome. Now, this is just a very easy question. Which blood type is the universal one? So it will be the universal donor is O, and the universal recipient is so AB can receive blood from everybody, and O can donate blood to everybody. And here, so here you can see this table. Here are the recipients, and here are the donors. So here you can see that O can donate to everybody, yeah, and B, so O can donate to everybody, while A and B, as you can see here, can receive blood from everybody. And this was an example of multiple alleles with codominance. Remember the blood type, and here you can see the universal donors and the universal receptor. Now the question 22 was also we talked about during the class. Which of the following terms describe the proportion of organisms whose phenotype matches the genotypes? And so if you remember, we talked about this in the class. We talk about this is the definition of penetrance. We talk about expressivity and penetrance, and the expressivity show a gradient. The penetrance, it was the one that the genotype, the phenotype matched the genotype. So for example, here, this is 100% penetrance. However, I show you that in other cases, they could be variable penetrance. So the answer here will be definitely penetrance. It will not be expressivity. Expressivity is when you have these gradients of variable expressivity, when you have these different tones of purple in the explanation. Okay, so this is a very interesting question. Sex-linked genetically inherited traits can appear in both males and females, only in males, only in females, or result from premarital sexual intercourse. So definitely this is wrong. And A and B and C. So remember that in humans, we have X and Y chromosome. Females will be X and X, and males in human will be X and Y. So you can have as you can definitely have diseases that can be linked to the Y chromosome, or diseases that they can be linked to the X chromosome. So the answer is A. So it can appear in both males and females. And here you can see an example of both the X-linked trait when we have a carrier mother, that is the one that we described, but in class with the color blindness and with the other examples. However, you can also have X-linked chromosomes, and you can see here the effects of crossing a father that is affected with a mother that is not affected. And it's again the same, you just have to make a Punnett square. And in that way you can find this different type of inheritance. So the answer definitely could be, definitely will be that both X and Y chromosomes can develop these sex-linked traits.